Thank you, Dr. Go again. My name is Chia Yean. I am the application specialist from Chemopharm, okay, Life Science Division. And thank you for joining the workshop today. And the topic that I will want to go through with you guys will be high throughput gene expression and genotyping with microfluidic technology. And with micropower, you gain mega insights, right? So a brief introduction on Chemopharm. We are a group of companies distributing instruments from analytical, from life science, oil and gas, healthcare, service, and also chemolinks. And I'm actually from life science division, and these are the brands that we are distributing. Okay, the major brands are like Cytiva, Tcant, Integraph, Fluidigm, and etc. Okay. And just a brief introduction on Fluidigm Corporation. It is a US-based company, and it's specializing in microfluidic technologies. So it will be part under here. Just let me put up the pointer. Okay, so microfluidics, they are actually very strong in genomics and gene expression, especially for high throughput experiments. Okay, on top of that, they're also doing single cell genomics. So if you are maybe from cancer, doing cancer research, and you do want to see single cell gene expression or genotyping, then or even sequencing, then yes, it is. I mean, Fluidigm is one of the key vendor who manufacturing these IFC, the microfluidic channel for single cell genomic. Okay, and two more portfolio from Fluidigm is mass cytometry, which is the high parameter immune profiling. So I think you may heard of flow cyto. So mass cyto is another level higher. So like easily can be multiplexed up to like 40 parameters. There's no issue for that because it's using metal isotope instead of fluorescent dye. So that is mass cytometry. For imaging mass cytometry, it is still using mass cytometry. I would say it is more similar to it is like IHC, immune histochemistry. But immune histochemistry is normally you just see maybe one or two markers. But this one you can multiplex up to like 30, 40 markers. There's no issue because you are using a metal isotope. There's no crosstalk issue for either mass cytometry and imaging mass cytometry. Okay, then let me move on to a brief introduction. I think you should know by now what is gene expression and what is genotyping. So I will really go through very fast. So gene expression is the process by which information from a gene is used in the synthesis of a functional gene product. So we are using qPCR platform to detect gene expression. And there are two common chemistry being used in qPCR. So DNA binding dyes like cybergreen, evergreen, and also TaqMan probes from Thermo. Okay, at the end of the day, you will be getting this kind of graph, which is the amplification plot. If you're using DNA binding dyes, then you will get another piece of information, which is the melt curve analysis, right? So for SNP genotyping, it is the analysis of single nucleotide polymorphism, SNP, the short form, SNP, among individuals of a population. And it is one of the SNP genotyping. We can use PCR-based method to study SNP genotyping. So usually we are using two different fluorophores to determine the allele, FAM plus WIC or FAM plus HEX. Okay, and available chemistry option include TaqMan assay, which is from Thermo, as I mentioned earlier, and also SNP type assay, which is a proprietary product from Fluidigm. It's called SNP type assay. So I just wanted to touch a little bit more on the differences on SNP type and also on TaqMan. So TaqMan, we always say is probe hybridization. So on top of what you have as a forward and reverse primer, they are using a probe-based fluorescent tag to bind on the targeted site. Okay, while for SNP type assay, it is kind of like using the allele specific primer. So you can see the primers over here. This actually have this kind of like a short sequence that we are using universal probe. So for this kind of setup in SNP type, the cost will be much more cheaper comparing to TechMed Pro, because we are using universal probe for all types of for all the assays. So in, let's say, in in an experiment setup, you have, let's say you are screening maybe, let's say for example 10 SNPs. So all these 10 SNPs, they are using the universal probe, which is this one. Okay, but the primer has to be different. So that's very, very clear. So for overall SNP type assay, the cost is quite affordable at about 200 to 250 ringgit per pair of primers. Okay. And of course, the more data, which is better. So especially in SNP genotyping, you need to have really a lot of individual or subjects to to carry on the SNP genotyping experiments. Okay, it couldn't, for most of our customers, their SNP experiment is like a few hundreds, like can be like 100 or 200 of patient versus 100 or 200 of control cases. So it would be like a few hundred subjects in SNP genotyping experiments. For gene expression, it's depending. So sometimes they are looking at more genes so that they can cover more aspects. So they read on more genes. So that would be mainly from gene expression. So microarray is actually quite powerful, but anyhow, after the end of the experiment, you need to kind of like verify again with qPCR. But for Fluidigm, because it's already a qPCR based technology, so you do not need to revalidate again because it's already validated, right? So that is the message from this slide. Okay, what if you have this much of sample to screen? This is real case, all right? This is a real case from Alaska Fishery, I think Department of Fish and Game. So they are genotyping their salmon fish. So from here, do you want to guess how many plates they have in this photo? Do you want to have a guess? Although I don't have any free gift to give out, but would you want to guess how many plates they have been using to genotype their salmon fish? If you want, you can unmute yourself and let me know how close is the number to this the number of your guess with the number in these pictures. Do you want to have a guess? It's a 4-digit number. Okay, it's not 100, it's 1,000. Okay, so maybe I should I can announce the answer is about 1,003 plates, 3 A4 well plates. So they are really using this much of plates to genotype the salmon fish. Okay, and what if there is a solution for this where you can reduce the time, you can reduce the labor, and you can reduce the reagents? Okay, then yeah, I think everyone should be happy to have this kind of solution. Okay, can save your time, then can free up your time to do some other things, right? So yeah, so I want to talk more about the solution from Fluidigm and the technology behind it. Okay, and this is the setup from Fluidigm. So we have this Biomark, which is the qPCR platform, the high throughput qPCR platform. And then we have the consumables, which is this one. So to translate from the just now traditional qPCR plate into Fluidigm plate, we call it IFC, is actually just using 54 pieces of 96 format. I will explain later what is 96.96. All right, so you can see the big pool of plates if they are using Fluidigm, and actually they have switched to Fluidigm after them. So they use Fluidigm, and with that, with that, a lots of sample and also SNP to screen off, they are just using 54 plates of 96 format. Okay, so let me go further a little bit to the core technology. So it's actually the centerpiece of this integrated fluidic channel. So we call it, it's a very soft material like contactless material. So you can see there's more tiny little line over here. It is bringing the solution, the reagent from here into here, and all this small partition is happening at the middle part. Okay, so left side usually is for assay and right side is usually for sample. meaning assay number 1, meaning can be gene number 1, gene number 2, number 3, number 4, up to gene 96. This is what we need to do, pipette our primers in. And then on the right side will be our sample number 1, number 2, number 3, up to sample 96. Then the machine will help us to kind of like load and mix the sample. And these all happen in the middle of here, okay, in this reaction chamber in the middle. Okay, and you do not need to worry about the mixing and the loading. Is all done by the machine. Okay, what we need to do is just manual load on the inlet on the left and on the right side. Okay, we go a little bit deeper into this technology. Is actually thanks to this Nanoflex valve. So you can see here what I have circled over here. That is the kind of like intersection between control line and fluid line. So let me explain a little bit more on fluid line. Okay, fluid line is the line that carry our sample or our assays, can be our primers. Okay, control line is the kind of like a mineral oil from fluid line. So it is actually kind of like cross intersection with the fluid line. So whenever the pressure is applied, so the compartment here, okay, the area here will be collapsed, okay, because the material is soft, so it will collapse at the correct position. So how to put in the more layman term is like you're stepping on the garden hose, okay, or one leg, a left leg and a right leg. So you step on the garden hose. Is it middle part will be kind of like being partitioned. So that's how we explain in the layman term. So it's all because of this Nanoflex valve, right? And the material is soft, is a contact lens material. So whenever pressure applied, the position here will be collapsed, and here another intersection will be collapsed. So this will be one partition. So yeah, there are total like 9,216 partition max in a chip. So I will move on. So when we zoom in, like this is the view under a microscope. So yeah, samples. Sample, they are putting in food coloring, so you can see that there are some color over here. So yellow for sample, blue for assay. So you can see here, this is actually the valve. Okay, the valve. This is the valve to partition the assay. You can see here. Okay, while this is the valve where it will later be open and to mix two chamber. So this is assay, this sample, right? So after that, the valve will be open and then it will mix, okay, sample mix with our assay. So while this is the valve for sample, okay, this the Nanoflex valve for sample, Nanoflex valve for assay. All right, so I will move on to the next slide. And this is the video. You can see it is like, yeah, the yellow sample, colored sample will be pumped in by the machine, by the, we call it IFSC controller. It's all being done automatically. You do not need to worry about that. So pump in into the compartment. Okay, you can see here the valve is still closed. Okay. So sample going in first, yellow color. And then the assay going in, which is colored in blue. And you still notice that the Nanoflex valve over here is still closed. Okay, later on the Nanoflex here will be closed, will be how to say, will be pressed on, with pressure applied, and the nano valve here will be closed now. So now this is closed. Later on will be open for this compartment and this compartment to mix up. Okay, so this is loading scenario. I will move on to the next slide. Okay, now you can see the Nanoflex valve here is already closed, and you can see here will be opened up later. And this is mixing, a mixing process also being done by the machine, by the IFC controller. So that would be, I think that would be all of these. I think the video will stop here. Okay, let me move on to the next slide. Okay, yeah, to compare microtiter plates like 384 well plate or 96 well plate against Fluidine IFC. So this is the major differences lah. So for 384 well plate or for 96 well plate, we are usually mixing up one well will be one assay and one sample, right? So which means first sample versus first gene on the first well, then first sample versus second gene for the second well, the first sample versus third gene on third one, and etc. So it will be like this. Okay, user assemble each reaction by dispensing components differing by sample or assay in each well of microtiter plate. So this is the traditional qPCR plate. Okay. While for Fluidine, we have just now, I also explained a little bit, like we have like the assay inlet on the left side and the sample inlet on the right side. So these tiny inlets, we just need to put in our assay in, like primer number 1, targeted gene number 1, number 2, number 3, number 4, and etc. Sample on the right, assay on the left, and then the IFC controller will load and then mix up this assay and sample in the reaction chamber. Okay, and then not to worry about this tiny inlet. It is actually multi-channel pipette compatible. So you can see there are total 16 inlets over here, which means the odd and even. Okay, the odd position, one time of aspiration. Okay, you can load into position number 1, 3, 5, 7, 9, and etc. So the second time loading, another multi-channel 8-channel multi-channel pipette. So you can load into it, and then it will aspirate into well inlet number 2, 4, 6, 8, 10, and etc. So meaning to complete the loading for the assay inlet, you need for this particular format lah, 1, 2, 3, 4, 6 loading. 1, 2, 3, 4, 5, 6, 6 loading. You will complete all 48 wells over here. Another 6 aspiration, then you will complete the sample inlet. So it's very convenient. Okay. So for Fluidine IFC, user dispense assay and sample mix into designated inlets of the IFC, and then the system will assemble the reaction for us. Okay, and then controller automates the reaction assembly at the nanoliter scale at the middle here. Okay, and then second advantages comparing Fluidine IFC against microtiter plates. I would say Fluidine IFC really save up a lot of reagents and sample. And yeah, this is the example that I would want to show you. For example, let's say you have 96 sample and you have 96 gene to screen. For 384 well PCR system, you need roughly about 23 ml of master mixers, 2.3 ml of primer probes, 24 pieces of 384 well plates. And let's say one day you run 3 plates, then you need 8 days to complete the experiments, and you will be able to get 9,216 data points. So this data is actually come from 96 times 96, okay, 9,216 data points. So for Fluidine, okay, by using biomark system, yeah, you can still get back the data, the same data point, 9,216 data point, but you only need 240 microliter master mix, 240 microliter primer probe, 1 chip, which is 1 piece of 96.96 IFC, and you will be able to get it done within a day. Okay, some experts they did it like within 4 hours, but I would say to be on the safe side, I would say like a day, like 6 to 8 hours. Okay, and within a day you will be able to get up to 9,216 data points. Comparing to traditional PCR system, you need like a week time to get this pool of data. Okay, to summarize, Fluidine IFC advantages: neutralize the reaction, roughly about 2.5 or less than 5 microliter master mix for 96 sample. Okay, ultimate assembly of reaction with the help of IFC controllers. Reduce error with less hands-on processing. The more sample or the more plates that you handle, then it could have more errors lah. So that's the one of the advantages on using Fluidigm IFC to reduce error with less hands-on processing. And then flexible and open formats means the user is in control of content. Okay, and next, these are the formats that are available from Fluidigm, from medium throughput to high throughput. Okay, you can see Flex 6 IFC, they are 48.48, 96.96, and 192.24. So the number meaning the number of assay and the number of sample. Okay, number of sample, number of assay. So which means for this particular chip, it can screen up to like 48 genes against 48 sample. And this is 96.96, meaning 96 sample, 96 gene or 96 SNPs. And 192.24 is 192 sample against 24 genes or 24 SNPs. Okay, sample over here, and then SNP or gene on the left side here. Okay, well, for this one is left side assay, right side sample. Assay on the left side, right side of the right side for the sample. Flex 6 is a bit special because it has, why it's called Flex 6, because it's there are 6 partitions. So you can see there's one, the first partition over here, the second, the third, fourth, fifth, and sixth. So at one time, you can use a single partition, meaning open one partition for assay and one partition for sample. Then load here, 12 sample, 12 assay, and then let the controller mix up using up one chamber. So maybe next week you have another pool of sample that you want to carry on the gene expression studies, then yeah, you open up the second partition. Okay, then the third week, maybe you have another group of sample, then yeah, you open up the third chamber, the third partition to use. So that's why it's called like flexible 6 chamber. For the rest of the format is one-time use. So you need to like just pull more sample and then just run it at one time. Okay, you're able to get the data in within a day. Yeah, providing mid to high throughput scalability. 144 is this one, the most smallest scale among the IFC family. Okay, 144, meaning one partition. Okay, and then up to 9,360, which is this one, 96 times 96, you will get 9216 version. Okay, and then yeah, consistent and reliable data quality, and all without changing technologies or instrumentation. It's all using the same biomark system. Okay, just the different on IFC control. And for the reagents, it has you will always package in together. Yeah, there are control lines. Remember, there are 2 lines, right? Control line and fluid line. So this is the control line. This one, the special mineral oil from Fluidigm. Then we have this DNA binding dye sample loading reagent. We have assay loading reagent, and also RT, okay, reverse transcription master mix. And yeah, for the setup, you can see here, this is the qPCR platform. We call it Biomark HD. Okay? And then for the controller, we have 2 version controller. The older, not previous model. The simpler version controller is this one, IFC controller, just for IFC priming and loading. While for Juno is also for cycling, meaning if you are to perform SNP genotyping, because SNP is endpoint study, it's not a real time. So you may want to use Juno to help you to prime and then load and then mix our chip, and also cycling in this machine. And then you transfer the chip to Biomark to read, like within 10 minutes, you're able to read the plate. Okay, and then the third component will be the chip itself, IFC, depending on throughput, Flex 6 or 96.96, that's the different in throughput, and also the reagent. All right, so this is the setup lah. This is the things that you need to run using Fluidigm technologies. And for the workflow, yeah, first we are to do specific target amplification. This is just for those sample that which has lesser concentration that required. So for human sample DNA, let's say it is lesser than 60 nanogram per microliter, then we will encourage customer to do this specific target amplification. Then yeah, we prime the chip into on this IFC controller, and at the same time do some preparation, dilution, and etc. And then after that, we can transfer our sample and our assay mixes into the inlet over here. Okay, and then we transfer this chip into IFC control again to do the loading and the mixing. And then once done, like after an hour plus, then we transfer the chip to this biomark to read, okay, as a qPCR or SNP genotyping, still using the same machine, okay, biomark HD. Okay. For new setup, not new, for another setup, let's say they have plenty of chip to run and they do not want to use up biomark for thermal cycling and for reading, they can utilize this Juno system. So the first portion, the first 3 steps still the same, but for the fourth steps, they can load and thermal cycling in Juno, and then they can scan using the they can just scan using the biomark because the detector is on the biomark, not on the Juno. Okay, and I think I am almost to the end. Chemistry option for Fluidigm, it is actually open to TechMan and Evergreen. Okay, for gene expression, for SNP genotyping, it is also compatible with SNP-type and also TechMan. Okay, TechMan is from Thermo. Okay, SNP-type is from Fluidigm. Okay, and is compatible with most common chemistry in the market. And primer probe design, yes, we do have that. So you can just go to the Fluidigm D3, D3D design, define, design, deliver. So D3 assay design. Okay, just load on, create account, then yeah, just put on the either the RS number for SNP genotyping or the gene name for the gene expression. Okay. So yes, we do have primer and probe design and synthesis. Software, I think you also quite familiar, amplification plot, melt curve. Then we do have the heat map because sometimes if you wanted to look at the overall expression of your the whole project, then yes, heat map maybe is quite interesting piece of data or display for you to see. Okay, amplification plot and melt curve is quite standard for gene expression study. And for SNP genotyping, then you have these different colored dots. Okay, it can be like homogeneous or heterogeneous population, and you have this scatter plot. Okay, homozygous sample and then heterozygous sample, and those are not amplified sample. Okay, so quite common in SNP genotyping experiments. And publication, do not need to really worry too much. Okay, you can just go on the Google Scholar, type in Fluidigm, biomark, gene expression, or genotyping, then yeah, you have thousands of publications. Or if you want, maybe you are looking for some special application and you want to know more, then feel free to talk to us. Okay. And I think that's all from my side. Thank you. And do you have any questions? If you have, then feel free to unmute yourself and ask. If shy, then you can send email to us. My personal email is cyhong@chemopharm.com.my, or you can send to our group email in case I am away or not available. So yeah, my team member will help me to guide you through. Okay, life science team at chemopharm.com.my.